Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Imagine a master disguise, like a secret cloak of invisibility that some of the most formidable invaders, cancer cells, use to hide from our body's elite security force, the immune system.
Right.
Now, what if we could take that exact same cunning trick and flip it on its head?
Yeah.
What if we could use it not to hide something harmful, but to protect something incredibly vital, like the insulin-producing cells in your pancreas from a, devastating autoimmune attack?
Yeah.
Sounds like something out of a futuristic medical thriller, doesn't it?
It really does. Science fiction becoming fact, perhaps.
Well, today we're taking a deep dive into precisely that kind of groundbreaking science. We're exploring truly fascinating research from the Mayo Clinic that unexpectedly blurs the lines between cancer biology and a potential new life-changing treatment for type 1 diabetes.
It's a remarkable connection, absolutely. And for those of you who might be less familiar, type 1 diabetes is a chronic autoimmune condition.
Right.
In simple terms, your own immune system, which is, designed to protect you from invaders like viruses and bacteria, mistakenly turns on itself.
Attacks your own cells.
Exactly. It specifically targets and destroys the beta cells located in the pancreas. And these beta cells are absolutely critical because they're the only cells in your body that produce insulin.
The hormone essential for blood sugar regulation.
Precisely. Without functioning beta cells and insulin, the body just can't manage its blood sugar levels effectively, leading to a lifelong reliance on external insulin injections or pump therapy.
So our mission for this deep dive is really to unravel the intricate details of this study. We want to understand just how this sugar layer, as we're calling it, could be the unexpected key to protecting those vulnerable beta cells. And importantly, what this potentially revolutionary discovery could mean for the, estimated 1.3 million people in the United States currently living with type 1 diabetes. We'll explore why this research is causing such a ripple of excitement in the scientific community and what its long-term implications might be for treatment.
Yeah, and the impact of type 1 diabetes, well, as you know, if you or someone you care about lives with it, it's profound. It extends far beyond just the daily injections.
Absolutely.
Without diligent management, poor blood glucose control can lead to immediate and acute medical emergencies, like diabetic ketoacidosis, which is genuinely life-threatening.
Very serious.
And long-term, the sustained high blood sugar can cause severe complications affecting vital organs. The heart, kidneys, retinas, even the peripheral limbs, leading to nerve damage and potentially amputations. It's a really significant public health concern.
It's growing, right?
It is. Its prevalence is increasing globally by about 3 to 4% over the last 3 decades, which really underscores the urgent need for, new solutions.
And while medical science has come a long way with insulin delivery, offering everything from advanced, long-lasting insulin formulations to sophisticated pump systems that try to mimic the body's natural insulin release.
They do try.
They do. But they're still management tools, fundamentally not cures. They help immensely, of course, but they don't perfectly normalize blood glucose levels, nor do they stop the underlying autoimmune attack itself.
No, the root cause is still there.
So the daily burden of monitoring, calculating, injecting, it remains constant.
Exactly. For some, there is a more radical approach, pancreatic islet cell transplantation.
Yes, the transplantation.
This offers the promise of a potential cure, theoretically freeing patients from that relentless cycle of daily glucose monitoring and insulin dosing. It actually has shown incredible promise for long-term blood glucose normalization, sometimes for years.
But there's a catch, isn't there? A big one.
A very big one. There's a formidable obstacle. See, to prevent the body from rejecting the donor tissue, which is, after all, foreign to the recipient's immune system, and to suppress the existing autoimmune response that destroyed the original beta cells in the first place.
You need to suppress the immune system.
Heavily. Patients must take powerful, lifelong immunosuppressive drugs. And this, unfortunately, comes with serious adverse reactions. We're talking a significantly increased risk of severe infections and even certain types of cancer, certain malignancies. So you're essentially trading one set of problems for another.
Which is a really tough trade-off.
A very tough trade-off.
And this is where the story takes that truly captivating turn, that eureka moment that, as you said, didn't even start in diabetes research.
Right.
The surprising origin of this breakthrough came not from the labs focused on the pancreas, but from those studying cancer. The Mayo Clinic team, led by Dr. Virginia Shapiro, was initially looking into how cancer cells manage to outsmart our immune system. They identified a specific sugar molecule, a kind of sugar cloak, that these aggressive cells strategically place on their surfaces.
It really is quite ingenious in a sinister way how cancer cells do this. They effectively coat themselves in a specific sugar molecule called sialic acid.
Sialic acid. Yeah.
Think of it like biological camouflage. This sugar coating helps the tumor cells appear as if they are not foreign entities, basically allowing them to evade the immune system's detection and destruction.
Sneaky.
Very. And the key player in creating this camouflage, the enzyme responsible for increasing the sialic acid on tumor cells, is called sialitransferase. 8 sialitransferase family, a member 6.
Okay, that's a mouthful. Let's maybe call it ST8CA6 for simplicity.
Yes, ST8CA6 is much easier.
Right, so ST8CA6 puts the sugar coat on cancer cells.
Correct.
And this is precisely where Dr. Shapiro's pivotal question, her flash of insight, came in. She looked at this cancer cell trick, where this enzyme, ST8CA6, helps an abnormal cell hide from normal immune response, and wondered, what if we could harness that same mechanism? Could this enzyme help a normal, healthy cell, like an insulin-producing beta cell, be protected from an abnormal immune response? Specifically, the destructive one seen in type 1 diabetes.
It's a brilliant conceptual leap.
It really is. This profound hypothesis shift was the catalyst that redirected their research entirely.
So, to put this hypothesis to the test, the researchers turned to a very well-established preclinical model, the non-obese diabetic mouse, or NOD mouse.
Okay, the NOD mouse. Why that model specifically?
These particular mice are really considered the gold standard for modeling type 1 diabetes because they accurately mimic crucial aspects of the human disease. Things like the genetic risk factors, the specific autoantigens that trigger the immune attack, and the precise mechanisms by which beta cells are ultimately destroyed. It's a very good model.
So it closely resembles what happens in humans.
As closely as we can get in a mouse model, yes. And it's also important to note that this enzyme, ST8-CIA6, the one that's so effective at camouflaging cancer cells, it's not naturally found in beta cells under normal circumstances. They don't usually make it.
Okay, so that's key. They don't normally have this cloak.
Correct.
So with that bold hypothesis in mind, the next step was, well, to see if it would actually work. What they did was, brilliantly simple in concept. They engineered the beta cells in these mouse models to actually produce the ST8-CA6 enzyme.
Right. They gave them the genetic instructions to make it.
In essence, they were giving these vulnerable beta cells the same sophisticated sugar coat that cancer cells use to go undetected, hoping it would protect them from the autoimmune attack instead.
And the results were truly impactful. I'd even say astonishing.
What did they find?
The engineered beta cells proved to be an astounding 90% effective in preventing the development of type 1 diabetes in the female non-obese diabetic mice over a substantial 300-day study period.
90% prevention. That's huge.
It's huge. To put that in perspective, nearly 60% of the control mice, the ones who didn't receive the engineered cells, developed the condition during the same time frame.
Wow, quite a difference.
A massive difference. And crucially, the beta cells themselves, the ones that are typically destroyed by the immune system in type 1 diabetes, they were beautifully preserved in the engineered mice. They were still there, functioning normally.
Okay, so the protection worked. But this brings us to a really vital point about the immune response itself, something highlighted by Justin Cho, who was the first author on the paper. He stated that even though the beta cells were spared, the immune system remained intact. Why is that so important?
It's absolutely key because it means the protection was incredibly specific.
Yeah.
It wasn't just a broad shutdown of the entire immune system, which, as we discussed with transplantation, would leave the body vulnerable to other infections or diseases.
Right. Avoiding that trade-off again.
Exactly. They specifically observed that the mice still had active B cells and T cells, perfectly capable of fighting off other threats. The ST8-CA6 enzyme seemed to generate a very localized tolerance.
Like diplomatic immunity just for the beta cells.
That's a great analogy. Diplomatic immunity just for the beta cells, without compromising the overall immune system's ability to defend the body elsewhere.
Okay, let's unpack this further, because understanding how this sugar coating works to create such specific protection is, well, it's truly fascinating. How does it tell the immune system to back off only in this one specific instance?
It really comes down to an elegant cellular conversation, a molecular handshake, if you will.
Okay.
The sialic acids, those sugar molecules generated by the ST8-ASCS6 enzyme, act as what we call ligands. Think of them as keys.
Keys.
And these keys fit into specific locks, which are a family of receptors known as Siglic receptors. Siglic stands for sialic acid-binding immunoglobulin type lectin.
Siglic receptors. And where are these locks found?
They're predominantly found on the surface of various immune cells. Now, here's the critical part. Many of these Siglic receptors are inhibitory.
Inhibitory, meaning they stop something.
Precisely. When they engage with sialic acid, when the key goes into the lock, they send a signal into the immune cell, essentially telling it to stand down or don't attack. It's like a biological do not disturb sign, aimed specifically at that immune cell interacting with the sugar-coated beta cell.
Fascinating. So it's not hiding, it's actively telling specific immune cells to back off.
It's an active modulation, yes. And within the islets, those crucial clusters of cells in the pancreas containing the insulin-producing beta cells, the researchers observed very specific and significant changes in the protected mice.
What kind of changes?
Well, first, there was a considerable reduction in the overall infiltration of immune cells into these islets. Fewer soldiers were entering the protected zone.
Okay, less inflammation overall.
Exactly. Moreover, the key destructive immune cells, the ones that are usually the main aggressors in type 1 diabetes, specifically a type called CD8-positive short-lived effector T cells and also T helper 1 cells, were significantly reduced in number within the islets.
So the really aggressive ones were kept out or reduced.
Right. And critically, there was also an increased ratio of T-bet-positive regulatory T cells to these effector T cells. Think of these regulatory T cells, or TREGs, as the peacekeepers of the immune system.
The good guys in this context.
The good guys, yes. Their increased presence helps to actively create a more tolerant, less inflammatory environment locally, right there within the islet.
So it's shifting the balance towards tolerance.
Precisely. And perhaps most significantly, the researchers found something really interesting about a type of cell called type 2 conventional dendritic cells. These are a major immune cell population within the islets.
Okay, dendritic cells. What about them?
In the protected mice, these dendritic cells had significantly lower levels of a powerful signaling molecule, a cytokine, called interleukin-12 subunit P35. Interleukin-12 subunit P35. Why is that important?
Because this particular cytokine is a known key driver of the inflammatory destructive immune response in type 1 diabetes. Lowering it seems to really dampen the attack.
Got it. So multiple layers of local control happening.
Yes, it's a multifaceted local effect. And this all circles back to that groundbreaking idea you mentioned, peripheral tolerance. This is a huge deal here, isn't it?
It really is. This isn't about completely wiping out the immune system's ability to recognize self, like in central tolerance. It's about creating tolerance out in the periphery, at the site of the target tissue.
So it's like teaching a specific guard dog, an immune cell, to be friendly with one specific delivery person, the beta cell, while still keeping a sharp eye out for everyone else.
That's a very good way to put it. And the study really supports this. They found that autoreactive B and T cells, those immune cells capable of causing type 1 diabetes, were actually still present throughout the protected mice's bodies.
Still there, but not attacking the beta cells.
Exactly. In fact, these protected mice even still developed other autoimmune conditions, like Sjogren's syndrome, which this NOD mouse model also happens to represent.
Interesting. So the underlying autoimmune tendency wasn't erased.
No, it wasn't. This clearly indicates that the ST8-CS6 enzyme didn't eliminate the immune system's overall capacity for autoimmunity. Instead, it created a highly localized truce, a protective zone, specifically at the site of the beta cells.
Just shielding the beta cells themselves.
Precisely. And telling proof. They could even overcome this protection by administering extra interleukin-12 subunit P35 from the outside.
Oh, that cytokine again?
Yes. Giving it back essentially reignited the attack, further underscoring its critical role in the protective mechanism they observed. Blocking it or its production was key.
So with all this incredible insight, I mean, what does this all mean for the future of type 1 diabetes treatment? This is where it gets really exciting for people living with this condition.
It really does. The most immediate and perhaps profound implication is the potential to completely revolutionize pancreatic islet cell transplantation.
How so?
Well, if beta cells can be engineered to be tolerated by the immune system in this highly specific way, using their own cloaking device, essentially,
Well,
it could potentially eliminate that critical need for lifelong systemic immunosuppression.
which would be a total game changer.
A complete game changer. It would make a potentially curative therapy, like islet transplantation, much, much safer and vastly more accessible. It dramatically reduces those severe risks of infection and malignancy that currently come with the immunosuppression.
That alone is massive. What else?
Well, what's also incredibly promising and offers a genuine beacon of hope is that the study showed this tolerance seems to be robust and remarkably long-lasting.
It sticks around.
It seems to. They found that if the expression of ST8-CS6, the enzyme making the sugar coat, was maintained past 20 weeks of age in the mice, the protection actually persisted even after they stopped the expression of the enzyme.
Really? So the immune system kind of learned.
That's what it suggests. It indicates that a stable, lasting immune tolerance was established. It wasn't just a temporary effect that vanished when the enzyme production stopped. The immune system seemed to have effectively learned to leave those specific beta cells alone long term.
That suggests potential for a more permanent solution, not just continuous treatment.
That's the hope, yes. A way to induce lasting tolerance.
Now, we have to be realistic, too. While this is incredibly promising, you mentioned it's still early stages, right? Preclinical.
Absolutely crucial to acknowledge that, yes.
Yeah.
It's still in early preclinical stages. The non-obese diabetic mouse model, while it's an excellent tool for studying type 1 diabetes, isn't a perfect replica of the human disease.
What are some key differences?
Well, for instance, there's a known sex bias in the mouse model. Female NOD mice are much more susceptible to developing diabetes than males. That kind of strong sex bias isn't typically seen in humans with type 1 diabetes.
Okay.
And, the translation of therapies from mice to humans can sometimes be imperfect just due to underlying physiological differences between species. What works in a mouse doesn't always work the same way in a person.
Standard challenge in medical research.
A standard, but very real, challenge. Also, a key point. This particular study focused primarily on preventing the disease in mice that were genetically predisposed but hadn't developed it yet.
Right, prevention versus reversal.
Exactly. Future research will be absolutely vital to explore if this innovative approach can modulate, or perhaps even reverse, already established autoimmune responses in individuals who have already been diagnosed with type 1 diabetes. That's often the situation for most patients seeking new therapies. Can it stop an ongoing attack or even help regenerate function? That's the next big question.
Huge question still to answer, but the potential is undeniable. We've seen how an unexpected, almost counterintuitive insight from cancer research is opening up entirely new avenues for treating type 1 diabetes. This deep dive truly showcases the incredible, interconnected power of scientific discovery, where understanding one complex biological mechanism can, quite surprisingly, unlock solutions for seemingly unrelated diseases.
It really does. And this research critically highlights the importance of understanding these subtle immune checkpoints, like the sialic acid interactions we discussed.
These molecular handshakes.
Exactly. It powerfully demonstrates how precisely targeting the immune response at a very local level, creating that specific peace zone around the beta cells, rather than broadly suppressing the entire immune system, could lead to far more effective and, crucially, much safer treatments. Not just for type 1 diabetes, potentially, but maybe for a whole range of other autoimmune conditions, too.
That's a really exciting prospect. Localized immune tuning instead of system-wide suppression.
That's the goal, precision immunology.
So, final thought for you, our listeners, to consider. How many more unexpected connections like this lie hidden within the vast, intricate tapestry of biological systems? How might continued, bold, cross-disciplinary research looking at cancer to solve diabetes, for instance, continue to reveal these surprising links, leading to breakthroughs we can barely imagine today, ultimately improving human health in profound and truly transformative ways? It's a powerful reminder, I think, of the boundless joy of discovery and really the fundamental interconnectedness of all life.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.